All right, ladies, we'll give everybody a couple more minutes to come in, and then we'll pick up where we left off. All right, we're still waiting for one or two more people to come back. All right, looks like we just about have everybody here. So I'm going to go ahead. This is the slide that we looked at right before we went onto a break. And again, this is the way that we interpret an OF glucose test. And remember, we put glucose in primarily because this isn't about figuring out all the different sugars that a microbe can make use of. Most of our microbes are using glucose at least. So we use that in the OF test because of the fact that everybody can make use of it. If you start over on the left-hand side, remember, we're looking for a color change, and we need to figure out whether or not that's happening in air, aerobically, or under oil, or both. So again, when you see that yellow color in the media, that tells you that the pH must have dropped down. You must have an acidic product made. And it's the acidity that's turning that dye yellow, just like we saw in our fermentation tests last week. So in this pair of tubes, in part A, we see that yellow color in both. So this is an organism that's both oxidative and fermentative. This organism is oxidative because it's turned the media yellow in air, but it's not fermentative because the media has not turned yellow under oil. And over here, we have an organism that didn't use the glucose at all, essentially, because we don't see that yellow color in either tube. All right, now I want to mention just one important thing about this OF test that can be confusing when you think about it next to carbohydrate fermentation. And this goes back to when I said you have to know what indicator dye you're using. Well, you also have to know what kinds of acids are being produced that will turn an indicator dye yellow. So remember, when we looked at fermentation last time, we said the kinds of acids that get produced during fermentation, and remember, fermentation can produce acids, it can produce alcohols, and it can produce gases. And when acids are produced, those acids drop the pH down and they turn the media yellow. And if you remember, we said, depending on the fermentation pathway, you can get all different kinds of acids made, but they're all acids. So lactic acid and propionic acid and butyric acid, there's lots of them that can be made during fermentation. As long as it's an acid, though, it turned the fermentation media yellow. Now, in this OF test, we also see the media turning yellow? It's a different dye, it's a different indicator dye, but it still turns yellow in the presence of acid. So we have to ask ourselves, we know where acid comes from in fermentation. It comes from, lactic acid, propionic acid, butyric acid. But the question is, where does the acid come from in cellular respiration? We don't talk about cellular respiration in terms of making an acid somewhere, but it does. Remember the pathway all the way through. When you start with glucose and you go through cellular respiration, first it's converted to pyruvate, and then that pyruvate gets converted to acetyl-CoA, and then the acetyl-CoA gets converted to carbon dioxide. Now, carbon dioxide is not by itself an acid. Carbon dioxide does, however, react with water and become an acid. In fact, most of the acid inside the human body is the result of carbon dioxide becoming carbonic acid, or carbonate is the other word that we talk about. Both of those terms mean the same thing. So yes, when you're looking at the OF test and you see a yellow color change in the open tube, the tube that's exposed to air, we say, oh, this is the oxidative tube. This is the tube where cellular respiration is going on, the open air tube. The reason the media turns yellow in that tube is because there's acid in there. The cellular respiration produced carbon dioxide from the glucose, and the carbon dioxide mixed with the water in the broth and created carbonic acid. So just a little something to know about with regards to where the acid comes from in those tubes. So we have to be able to evaluate both tubes. We have to be able to evaluate the tube that's open to the air, the oxidative tube, and the tube that's under the oil, the fermentative tube. All right, let's take a look at some results. So in your lab manual, if you go to page 113, that's the beginning of our results page. I'm going to let you fill out the purpose. For the hypotheses, there are two statements that you need to agree or disagree with. The first one says that all bacteria will use glucose, and the second one says that all bacteria will hydrolyze starch. Remember, we use glucose for a reason in the OF test. There's a reason that we choose that one as our carbohydrate, all right? Virtually every microbe is going to be able to use glucose. Now, I can't say that 100% because I don't know that to be true, but for the bacteria at least, glucose is used for the OF test because it's a sugar that all bacteria can use. Some of them are going to use it for cellular respiration. Some of them are going to use it for fermentation. Question 2 says all bacteria will hydrolyze starch. That one is not true because remember, that's the whole reason we do the starch hydrolysis test. We want to be able to see whether or not a bacterium can release its exoenzyme like amylase and break down the starch in its environment. That's a test we can use to distinguish microbes one from the other, whether or not they are capable of starch hydrolysis. So that brings us into the results section. And you can see in the first table for results, it's the starch hydrolysis results. And there are 3 organisms listed. And for once, those are the 3 organisms that we're going to have results for. So Bacillus subtilis, E. coli, and Pseudomonas aeruginosa. So you have three columns you need to fill out. You have the growth column, you have the color of the medium around that growth, and then you have whether or not the starch was hydrolyzed. So let's take a look. On the slide that you can see right now, you can see 3 plates. You can see the E. coli plate, you can see the Bacillus subtilis plate, and the Pseudomonas plate. Remember, when the plates come out of the incubator, they don't look like this. When the plates come out of the incubator, we have to put the iodine onto the plate, and then we'll get that dark blue, black color in the medium. It will turn that dark blue color wherever starch is present. And I know it's hard to see, but there is growth on all 3 of these plates. There is growth of E. coli cells on this plate. They're actually colonies, but they're hard to see. So this plate remained black. This plate is covered in starch. Okay, now compare that to the Bacillus subtilis plate. You can see where the organism was streaked onto the plate and where it grew. We got growth there. And you can see this area around the growth where the media turned this clear color. It still has sort of a brownish tint to it from the iodine, but it's clear. If you were to put a piece of paper behind that Petri dish, behind that agar plate, you would be able to read the writing in those clear areas. All right, so this is a plate where the starch was broken down, where the starch was hydrolyzed. So these cells, these Bacillus subtilis cells, must have secreted amylase to break down that starch. And then our last result is for Pseudomonas. You can see a straight line was streaked onto this plate. One straight swab line was streaked onto this plate. You can see that growth occurred on the Pseudomonas plate, but there are no clear areas here. No clear areas on this plate, no starch hydrolysis. All right. If you turn the page, if you go to page 114, at the top of the page, there's a table for our OF glucose. And the first thing it asks underneath that is the color of the uninoculated medium. And remember, that medium has a green color to it. So before we put the cultures in, the media has a greenish color to it. If acid gets produced in the tube will turn yellow. And if an alkaline product gets produced, like ammonia or something, the tube will turn dark blue. So we're going to stay with the same 3 organisms for these results: E. coli, Bacillus subtilis, and Pseudomonas aeruginosa. So you can see you're going to have to cross out a couple of organisms in your table. We didn't use Micrococcus luteus, so cross that out. And we're not using Alkaligenes faecalis, so you can cross that one out. And you can just add in Bacillus subtilis. So 3 organisms. And again, the first thing they ask about is growth. Don't worry about that. You can go ahead and cross out the growth area. I don't want to get into the specifics of which ones are aerobic and which ones are anaerobic. I'm more interested in the color changes. So you can cross out the part of the data table that asks about growth. Just know that we got growth in each of the tubes that we're going to interpret. The next column over says color, and you can see it's split into the aerobic tube column and the anaerobic tube column. So look at the Bacillus subtilis tubes first. Here's the uninoculated tube. Here's the control tube. So this tube had no organisms put into it, and there's that green color. Now, they've got this marked K for alkaline. Don't worry about that. I'll talk about that in just a minute. Just know that the green, this greenish color is sort of the uninoculated color. Now, Bacillus subtilis has a greenish color in the tube that's open to air and a greenish color in the tube that has oil over it. So you can record that for Bacillus subtilis as green in aerobic and green in anaerobic. Now, if we go all the way over to the right, these are the two tubes for E. coli. For the open tube, the aerobic tube, we have a yellow color, very obviously yellow. And for the oil-covered tube, we have a yellow color as well. So the results for E. coli are yellow, yellow. Now in the center, this is the Pseudomonas pair of tubes. In the aerobic tube, we have an interesting looking result. We have green color at the bottom of that tube and yellow color at the top. So for that result, for Pseudomonas aeruginosa, in the aerobic tube, you can write yellow/green. Yellow at the top, green at the bottom. And then for the anaerobic tube, the one under oil, you can see that the color remained this greenish color. So let's think about this for just a minute. Look at the E. coli tubes first. So E. coli, as you know, is a very interesting organism in that it is capable of living in a whole lot of different environments. We've used it all semester long, and we've seen that it can grow under all different kinds of conditions. It's very adaptive. And E. coli is classically known as a facultative anaerobe, which means it is capable of doing both oxidative metabolism, cellular respiration, and fermentation, and it does both very well. So both of these tubes turned yellow. There's acid in both tubes. It's just that the acid in the tube is different one to the other. In the aerobic tube, in the one that's exposed to the air, we know that respiration is going on in here. And remember, in respiration, carbon dioxide gets made, and that carbon dioxide can interact with water and create acid, create carbonic acid. In the oil-covered tube, the fermentative tube, this acid could be any of a couple of acids. It depends on the fermentation pathway that E. coli organism was using. So remember, it might be lactic acid, or it might be propionic acid, or any of the other acids that we talked about for fermentation. So this organism is capable of both oxidative and fermentative metabolism of glucose. So it got a positive on the OF test. The Bacillus subtilis looks like it didn't do anything, right? It looks like it didn't use the glucose in either tube. And this is where we kind of have to put a star on our results, because these particular pictures, these particular images are a little bit deceiving. I asked you to record them as green, green, and that's fine for our purposes. But I want you to know that there's an issue with the color in this image. And it's not that Bacillus subtilis can't use glucose. That's not true. It's just that the product that it makes has not turned this indicator dye into the correct color. I'll just say that. It's not that Bacillus subtilis doesn't use glucose. It does. But go ahead and record this as green, green. And the way that we would interpret that according to these results, is that it didn't do oxidative metabolism and it didn't do fermentative metabolism. Neither one, according to our results. Now look at the Pseudomonas. The Pseudomonas is kind of interesting. You can see that it didn't do any fermentation, again, according to our results. The media stayed green. But we got this mixed results in the aerobic tube. And this is actually something we see quite commonly in the OF test. Remember that depending on the microbe, cellular respiration can be done aerobically and it can be done anaerobically. Some microbes are strictly aerobic though. They can only use oxygen to do cellular respiration. Just like you and I require oxygen for us to do cellular respiration. And what you're seeing in this tube is cellular respiration is going on in here. But because it's an aerobic cellular respiration, you're only going to see the acid production. You're only going to see the yellow color to the depth that the oxygen can penetrate, to the depth that the oxygen can penetrate the media. So when you get that partial yellow, partial green color, it gives you an indication that respiration in this tube is completely oxygen dependent. And that this organism is, again, doing cellular respiration, but it's only doing it where oxygen is saturating into that broth. So we have 3 different organisms and 3 different results. And you'll have to go through as usual and think about conclusions. First of all, think about the hypothesis that we laid down at the beginning. We said that everybody uses glucose, but not everybody can hydrolyze starch. So you'll have to ask yourself some questions regarding those hypotheses. And then there's a couple questions below that for you to work through. When you get over to page 115, where it says critical thinking, there's only one question there that I want you to try, and that's question number 1. So for critical thinking, go ahead and try question number 1, but I want you to cross out questions number 2, 3, and 4. We're going to skip those. And then for the clinical application at the bottom, I want you to try that. I want you to try that question. Okay, if there's only one, and I think you might be able to work through that one just fine. All right, so using the chat, let me know if anybody has any questions about what we just talked about. Remember, I'm going to, as always, I'm going to post this slide set up on Canvas for you. Remember, you have to click on the lab course, though, not the lecture course. Click on the lab course and go into the modules section, and you will find these slides. I'm also going to try to post as much as I can. I'm going to post these Zoom meetings. Okay. Here's one question from Hannah. It says, none of the organisms use peptone, correct? That's the way to interpret this. According to our results here, we don't see anybody using peptone. And that's because if they were using peptone, then the media would have turned a blue color. And it's a very dark blue color when you see it. It's not easy to tell from the green, though. It's not nearly as blue as we saw, for example, in our citrate media. But That's the way you interpret that, Hannah. You're correct. If one of these organisms had not been able to use the carbohydrate at all, and it had used the peptone instead to survive, you know, as a nutrient, we would have seen some ammonia produced, and the indicator media would have turned blue. All right. So you should interpret that as right, according to our results, that they didn't use peptone. What I was trying to say about the bacillus is that this is not a normal result for bacillus. It's what we got. It's what I want you to record as our results. But this is not what we normally expect to see with bacillus. Remember, if you go back and look at our starch hydrolysis results for bacillus, bacillus is capable of hydrolyzing starch. Remember why an organism would do this. An organism would hydrolyze starch in order to get the glucose. So we know that Bacillus subtilis uses glucose. We know virtually all bacteria use glucose. So it's just not showing up in our result here. Oh, and the other question I have is about your pathogen papers. I'm hoping to get to your first draft pathogen papers later this week. I have a block of time set aside on Friday for that, and assuming nothing else comes up that disrupts that, I will be going through your first drafts on Friday, and then I'll get those back to you. Okay, good question. Any other questions? No hurry. Yeah, no hurry. I'm sure they're good. I'm sure you guys did fine on them. No worries about that. All right. I just want to encourage you to try as much as you can to try to stay up with the questions in your manual. I know it's hard. I know you have a lot on your plate right now, but as much as you can stay up with the labs and answer the questions as we go, I think you'll have a better time of it. And then you'll get, 5 out of 5 on the lab. So I'm going to encourage you to do that. And remember that when we meet, for example, on Wednesday, if you have questions about what's in the lab manual, jot it down, and we'll talk about it on Wednesday after you've had a chance to think about these questions for a day or two. We can certainly talk about the lab manual questions together, because I want everybody to have the best chance to get those correct and get those 5 points on each one of those. All right, so when we get together on Wednesday afternoon this week, we'll be moving on to the next lab exercise, which if you turn past fermentation, you'll see is protein catabolism. We're actually going to do 2 different days on protein, I think. I'll have to check the schedule again, but we're going to do protein catabolism next. So that's what we'll be talking about on Wednesday afternoon. Okay. Any other questions? All right. Remember, the discussion board is still there. If you have any questions as you're looking at the lectures or any of the slides or anything like that, everything is being posted up on Canvas for you. And if there's no other questions, I will see you on Wednesday afternoon. All right. Thank you, everybody. Thanks for coming. And we'll see you next time.